today our topic is immunology and immune response to cancer. So immunity and immunopathology are proverbial to edge of double-edged sword. So immunity is basically a double-edged sword. That is, it is a defense mechanism, but it can be injurious to the human body in a variety of ways. So it is two types: innate immunity, also called natural or native immunity; adaptive immunity, also called acquired or specific immunity. Innate immunity, natural or innate immunity is non-specific and is considered as the first line of defense without antigen specificity, and this innate immunity present from our birth. Intrinsic mechanisms that are poised to react immediately constitute the first line of defense. It is mediated by cells and molecules that recognize product of microbes and dead cells, induce rapid protective host reactions. Innate immunity is always present, ready to provide immediate defense against microbes and to eliminate damaged cells. The receptor and components of innate immunity have evolved to serve this purpose. So what are the components of innate immunity? The major components: epithelial barriers that block entry of microbes, phagocytic cells, mainly neutrophils and macrophages, dendritic cells, natural killer cells. These are the main components. Epithelial skin and GI system and respiratory tract act as mechanical barrier to the entry of microbes from the external environment. Monocytes and neutrophils are phagocytes in the blood that can be rapidly recruited to any site of infection. Monocytes that enter the tissue and mature are called macrophages. Then dendritic cells are specialized cells present in epithelial, lymphoid organs, and most tissues. They capture protein antigens and display peptides for recognition by T lymphocytes. Innate lymphoid cells are tissue-resident lymphocytes that lack T-cell antigen receptor and cannot respond to antigen, but instead are activated by cytokines and other mediators produced at site of tissue damage. Other cell types. Several other cell types can sense and react to microbes. These include mast cells, which are capable of producing many mediators of inflammation, and even epithelial and endothelial cells. Plasma proteins. In addition to these cells, several soluble proteins play an important role in innate immunity. The complement system consists of plasma proteins that are activated by microbes. Complement activation may occur through alternative and lectin pathway as part of innate immune response or through the classical pathway. Now, adaptive immunity. The adaptive immune system consists of lymphocytes and their product, including antibodies. These are of two types: humoral immunity and cell-mediated immunity. Humoral immunity, which protects against extracellular microbes and their toxins. Cell-mediated immunity, which is responsible for defense against intracellular microbes and against cancers. It develops more slowly than innate immunity, but is even more powerful. Humoral immunity is mediated by B, bone marrow derived, B lymphocyte. B lymphocyte, B stands for bone marrow derived. And there are secreted products, antibodies, also called immunoglobulin. Cellular immunity is mediated by T lymphocyte. T for thymus derived. One is bone marrow derived and another is thymus derived. Cells of the adaptive immune system. Mature lymphocytes that have not encountered the antigen for which they are specific are said to be naive, immunologically inexperienced. After they are activated by recognition of antigens and other signals, lymphocytes differentiate into effector cells, which perform the function of eliminating microbes. Memory cells, which live in a state of heightened awareness and are able to react rapidly and strongly to combat the microbe in case it returns. That's why it is called memory cells. So, these are the function of antibodies. These are the function. B lymphocyte, neutralization of microbe, phagocytosis, complement activation, activation of macrophages, inflammation, activation of activation and differentiation of T and B lymphocyte, and cytotoxic T lymphocyte, killing of infected cell, and regulatory T lymphocyte function is suppression of immune response. These are the different types of lymphocytes and their main function. Now, tissue of the immune system. The tissue of the immune system consists of primary lymphoid organ in which T and B lymphocytes mature and become competent to respond to antigens, and the secondary lymphoid organs in which adaptive immune response to microbes are initiated. Primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs, the principal primary lymphoid organs are the thymus, where T cells develop, and the bone marrow, the site of production of all the other blood cells, including lymph cell. Secondary lymphoid organs, the secondary lymphoid organs, lymph nodes, spleen, and the mucosal and cutaneous lymphoid tissues. So there are two types of immunity, innate immunity and adaptive immunity. While normal function of immunity is for body defense, its failure or derangement in any way results in disease of the immune system. First is immunodeficiency disorder, are characterized by deficient or absent cellular or humoral immune function. This group is comprised by a list of primary and secondary immunodeficiency disease, including AIDS. Second is hypersensitivity reaction. Third is autoimmune disease, occur when the immune system fails to recognize self from non-self. Fourth is possible immune disorder like amyloidosis. In any discussion of immunity, a few terms and definitions are commonly used. One is antigen. Antigen is a substance, usually protein in nature, which when introduced into the tissue stimulate antibody production. Antibody is a protein substance produced as a result of antigen stimulation. So, what are the types of antibody? IgG, IgA, IgM, IgE, and IgG. Cells of immune system, these are the cells of immune system. Lymphocyte, monocyte and macrophages, mast cells and basophils, neutrophil, eosinophil. These are all are the cells of immune system. Lymphocytes is the master of human immune system. Morphologically, lymphocytes appear as a homogeneous group, but functionally two major lymphocyte populations, T and B lymphocytes, are identified, and a third type is there that is called NK cell or natural killer cell, comprised a small percentage of circulating lymphocyte. So T cell, T lymphocyte, B lymphocyte, and natural killer cell or NK cell. B and T lymphocyte proliferate into memory cell, imparting long-lasting immunity, while B cell differentiate into plasma cell which form specific antibody. T cell get functionally activated on coming in contact with appropriate antigen. Upon coming in contact with antigen, the macrophage and the major histocompatibility complex MHC in the macrophage determine whether the invading antigen is to be presented to B cell or T cell. Some strong antigen that cannot be killed by antibody response from B cells such as microorganisms such as virus, mycobacteria, tuberculosis, Mycobacterium leprae, cancer cell, tissue transplantation antigen are presented to T cell. Now B cell. These cells are involved in humoral immunity by inciting antibody response. B cell circulation comprises about 10 to 15% of lymphocyte. On coming in contact with antigen, B cells are activated to proliferate and transform into plasmacytoid lymphocyte and then into plasma cell. Common B cell marker CD19, CD20, CD21, CD23. T cell help to provide B cell by a subset of T helper cell, Th2, by elaborated interleukin, IL4, IL5, IL10 and IL13. T cell. These are implicated in inciting cell-mediated immunity and delayed type of hypersensitivity. T cell in circulation comprise 75 to 80% of lymphocyte. T cell markers CD3, CD7 and CD2. Depending upon functional activity, the major subtypes are T helper or CD4 T cell and T suppressor or CD8 plus cell. T helper cell abbreviated as TH cell, these cells promote and enhance the immune reactions and are also termed as T regulatory cells. So Th1 cell elaborate interleukin 2 and interferon, Th2 cell elaborate interleukin 4, 5, 6 and 10. T suppressor cell abbreviated as TS cell. They suppress immune reaction but are cytotoxic and actually destroy the invading antigen. Hence are also termed as cytotoxic lymphocytes, CTL. These cells carry CD8 molecule on their surface and hence are also called CD8 plus cell. Now natural killer cell. NK cell comprises of 10 to 15% of circulating lymphocyte. These lymphocytes do not have B or T cell marker, nor are these cells depending upon thymus for development, unlike CD4 and CD8 T cell. NK cell carries surface molecule of CD2, CD60, CD56. NK cells are part of natural or innate immunity. Then monocyte and macrophages. Circulating monocytes are immature macrophages and constitute about 5% of peripheral lymphocyte. They remain in circulation for about 3 days before they enter tissue to become macrophages. Now the important features of immune function of macrophage. First is antigen recognition. They possess cell surface receptor to several extracellular molecule receptor for cytokines, component of complement, selectin, integrin and Fc of antibody. Then phagocytosis, antigen that has been recognized by the macrophages due to availability of above-mentioned surface receptor or the opsonized antigen is ready to be engulfed by process of cell eating by macrophages. Then secretory function. Macrophages secrete important substances such as cytokines and prostaglandin, which are chemical mediators of inflammation and activate other lymphocytes. Secretion of proteins, acute phase reactants, example fibronectin, macroglobulin. Then number 4 is antigen presentation. When macrophages are unable to lyse an antigen on an organism, the next best course adopted by them is to act antigen presenting cell for presenting to immunocompetent T cell or to B cell. Then basophil and mast cell. Basophil and mast cell have IgE surface receptor. Thus on coming in contact with antigen binding to IgE, these cells get activated and release granules, that is they degranulate. These granules contain substances such as histamine, platelet activating factor, heparin. Cytokines, messenger molecule of the immune system. Many functions of leukocytes are stimulated and regulated by the protein called cytokines. Molecularly different cytokines are called interleukin because they mediate communication between leukocytes. Classification, hematopoietin family, GCSF, GMCSF, then interleukin 1 alpha and interleukin 1 beta, tumor necrosis factor, platelet-driven growth factor, and transforming growth factor family. Then chemokine family. These regulate movement of cells and act through G protein-derived receptor that is IL-8 monocyte chemokine protein mutation. Now, mode of action of cytokine. How cytokine works? So, there are three ways: autocrine, paracrine, and endocrine. Autocrine, when a cytokine acts on the cell which is produced it. Paracrine, when it acts on another target cell in the vicinity. Endocrine, when the cytokine secreted in circulation acts on a distinct target. Cytokines are involved in following actions. Number 1 is regulation of growth. Number 2 is inflammatory mediators. Number 3 is activation of immune system. Number 4 is cytokine storm. Now, major histocompatibility complex, MHC. The function of MHC molecule is to display peptide fragments of protein antigens for recognition by antigen-specific T cells. In human, the MHC molecules are called HLA, human leukocyte antigen, because they were initially detected on leukocyte. That's why it is called HLA. The genes encoding HLA are clustered on small segment of chromosome 6. MHC is of two types: class 1 MHC molecules and class 2 MHC. Class 1 MHC regulate function of cytotoxic T cell or CD8 plus T cell. And class 2 MHC antigen regulate the function of helper T cell, CD4 plus T cell. So MHC class 1 regulate function CD8 and class 2 MHC function regulate the function of CD4. Then cell-mediated immunity. Naive T lymphocytes are activated by antigen and co-stimulators in peripheral lymphoid organs and proliferate and differentiate into effector cells that migrate to any site where microbial antigens are present. The principal co-stimulator for T cells are the B7 proteins that are expressed on antigen-presenting cell and are recognized by CD28 receptor on naive T cell. This co-stimulator is important. One of the earliest response of CD4 plus helper T cell is secretion of the cytokine IL-2. This creates an autocrine loop wherein IL-2 acts as a growth factor that stimulates T cell proliferation, leading to an increase in the number of antigen-specific lymphocytes. The function of helper T cells are mediated by combined actions of CD40 ligand and cytokines. When CD4 plus helper T cells arise, antigen being displayed by macrophages or B lymphocyte, the T cell expresses CD40 ligand, which engages CD40 and the macrophages or B cell and activate B cells. Some of the activated CD4 plus T cells differentiate into effector cells that secrete distinct set of cytokines and perform different functions. Cells of the Th1 subset secrete the cytokine interferon gamma, which is a potent macrophage activator. The combination of CD40 and interferon gamma mediate activation result of classical macrophage activation, leading to the production of microbiological substance in macrophages and the destruction of ingested microbes. Activated CD8 plus T lymphocytes differentiate into CTL that kill cells harboring microbes in the cytoplasm. By destroying the infected cells, CTL eliminates the reservoir of infection. CTL also kills tumor cells by recognizing tumor-specific antigen. Now the humoral immunity. Activation of B lymphocyte. Upon activation, B lymphocytes proliferate and then differentiate into plasma cell, and that secrete different classes of antibodies with distinct functions. Antibody response to most protein antigens require T cell help and T cell dependent. In this response, B cell that recognize protein antigens by their immunoglobulin receptor endocytose these antigen into vesicles, degrade them, and display peptide bound to class 2 MHC molecule for recognition by helper T cell. The helper tissues are activated and express CD4 ligand and secret cytokines, which work together to stimulate the B cell. Now these are the functions of antibodies. Neutralization of microbes and toxins, obstruction and phagocytosis, antibody-dependent cytotoxicity, lysis of microbes, phagocytosis of obstructed microbes. Now the immune response to cancer. It has long been known that body's immune system can recognize tumor cells as non-self and they attempt to destroy them and limit spread of cancer. The following observations provide basis for this concept. Number 1 is certain cancer evokes significant lymphocytic infiltrate composed of immunocompetent cells such as tumors have somewhat better prognosis like medullary carcinoma of breast, seminoma testis. Number 2 is rarely a cancer may spontaneously regress partially or completely, probably under the influence of host defense mechanism. Number 3 is it is highly unusual to have primary and secondary tumors in the spleen due to its ability to destroy the growth and proliferation of tumor cells. Number 4 is existence of immune surveillance is substantiated by increased frequency of cancer in immunodeficient hosts. Assuming the immune system is capable of recognizing and eliminating nascent cancers, it follows that a tumor that grow out in immunocompetent individual must be composed of cells that are either invisible to the host immune system or that activate mechanism that suppress host immunity. The following session explores some of the important questions about tumor immunity. What is the nature of tumor antigen? Second is what host effector system recognize tumor cells? How do tumors evade this host mechanism? How can immune reactions against tumor be exploited therapeutically? First is tumor antigen. Tumor cells express surface antigen which have seen in animals and some human tumors. Number 1 is tumor-specific antigen located on tumor cells and an unique or specific antigen for particular tumor and not shared by normal cells. Second is tumor-associated antigen are present on tumor cell as well as on some normal cell from where the tumor originated. However, it is now known that TSA and TA can both be present on normal cell and categorization into TSA and TA does not hold true. Thus, presently, distinction of tumor antigen is based on the recognition by host immune cell, that is CD8 plus T cell, and by the molecular structure of the tumor antigens. Currently, various groups of tumor antigens, oncoproteins, number 1 is oncoproteins from mutated oncogenes, number 2 is protein product of tumor suppressor genes, number 3 is of overexpressed cellular protein, abnormally expressed cellular protein. The classic example is present of MAGE gene is silent in normal adult tissue except in male germline, but MAGE gene are expressed on surface of many tumors such as melanoma, cancer of liver, lung, stomach and esophagus. Then number 5 is tumor antigens from viral oncoproteins. Many oncogenic virus express viral oncoprotein which result in expression of antigens on tumor cell, that is viral oncoproteins of HPV in cervical cancer and EVNA protein of EBV Barrett's lymphoma. Then second is anti-tumor immune response. The principal immune mechanism of tumor eradication is killing of tumor cell by CTL specific for tumor antigen. This mechanism predominant because the majority of tumor neoantigens consist of mutated gene products of viral protein that are endogenously synthesized and presented in the context of MHC class 1 molecule, enabling their recognition by CTL. Then cell-mediated mechanism. This is the main mechanism of destruction of tumor cell by the host. The following cellular response can destroy the tumor cell and induce tumor immunity in humans. Specifically sensitized cytotoxic lymphocyte, that is CD8 plus T cells, are directly cytotoxic to target cell and require contact between them and tumor cells. Then natural killer cell, a lymphocyte which after activation by IL-2 destroy tumor cell without sensitization either directly or by antibody dependent cellular cytotoxicity or ADCC. NK cell together with lymphocyte are the first line defense against tumor cell and can lyse tumor cell. Then macrophages are activated by interferon gamma secreted by T cells and NK cells, and therefore there is close collaboration of these two subpopulations of lymphocyte and macrophages. Then humoral mechanism. As such, there are no anti-tumor humoral antibodies which are effective against cancer cell in vivo. However, in vitro humoral antibody may kill tumor cell by complement activation or by antibody-dependent cytotoxicity. Then immune regulatory mechanism. Most ulcers grow relentlessly in spite of host immunity. This is explained due to direct controlling mechanism of immunity, or as under during progression of the cancer, immunity cells may disappear, cytotoxic T cells and NK cells may play a self-regulatory role, immunosuppression may take by various acquired carcinogenic agents. Now the cancer immunotherapy. Current status of immunotherapy is focused on following three main approaches. The first approach is cellular immunotherapy consists of infusion of tumor-specific cytotoxic T cell, which will increase the population of tumor-infiltrating lymphocytes, TIL. And the tumor which produces more TIL, the prognosis of that tumor is good. The patient's peripheral blood lymphocytes are cultured with interleukin-2, which generate lymphocyte-activated killer cell having potent anti-tumor effect. Then, second is cytokine therapy is used to build up specific and non-specific host defense. These include interleukin-2, interferon alpha, interferon gamma, tumor necrosis alpha, and GM-CSF. Then, monoclonal antibody therapy is undergoing trial against CD20 molecule of B cell in certain B cell leukemia and lymphoma.